Hello and welcome to the video lectures for Chapter 2 in EFB103. I am Dr. Chris Whips. Let's get started. So this chapter is all about the chemical context of life. The three key topic areas are listed below: atomic structure, chemical bonds, properties of water. We'll go through all of these. And sometimes we think about, well, why chemistry? Why do we need to start there? Well, we can work our way one direction or the other. We can think of biology as the application of chemistry. Every living thing is made out of chemicals, and how those living things work depend on those chemical interactions. We can go a little bit further back. Chemistry is dependent on physics, so the properties of those atoms that make up chemicals, and then ultimately subatomic particles. And so we'll start with atomic structure. Something else that might come up that you might hear or have heard, this idea of, well, I don't want to eat something because it has chemicals in it, or I'm going to buy this food because it doesn't have chemicals. Well, remember, everything is made out of chemicals. All living things are, and everything is made out of matter. Matter is anything that takes up space and has mass. And so sometimes chemicals, quote unquote, are used to describe maybe things that are undesirable. But remember, everything is made out of chemicals. And there are certainly some kinds of chemicals that are dangerous or toxic. But there's another point, a little bit of an aside here, that it depends on how much of those chemicals are in things. So this concept of the dose makes the poison. Pears, for example, contain formaldehyde. You can't just go drinking a gallon of formaldehyde, but the teeny tiny amount of formaldehyde that's in pears is not harmful to you. So that idea of dose makes the poison. Everything is made out of chemicals. And of course, chemicals are made out of atoms. So let's look at atomic structure. So atoms are made out of protons, neutrons, and electrons. Protons are the positively charged particles. Neutrons are neutral. Those form the nucleus of the atom. And then electrons swirl around that. And those are these negatively charged particles. Here in this image here, we see electrons buzzing around this central nucleus. Another way to visualize that is in this figure here. See, here we have hydrogen and oxygen. You see the nucleus composed again of protons and neutrons. In the case of hydrogen, just the one proton. And then you see electrons swirling around that nucleus. It is interesting if you think about the scale. So we look at a cartoon like this, and the nucleus looks really big. And that's helpful for our understanding and visualization of what an atom might look like. But if we put it on a different scale, let's say on the scale of the carrier dome, or the stadium as they're now calling it, how big, if the circumference of that atom with the electrons was the size of the carrier dome, how big would that nucleus be? If we look at our cartoons, the nucleus seems to take up a lot of that space. In reality, depending on the atom, of course, that nucleus might be the size of a softball in the middle of the field. So very small. And this is kind of mind-boggling, really, that atoms really are a lot of empty space. All right, so every atom has a characteristic, let's say number of protons. Not that every atom has a different number of protons. If they have the same number of protons, they are the same element. And this number of protons is equivalent to the atomic number. So you got the same number of protons, that's going to tell you whether you're dealing with oxygen or carbon or whatever. Same number of protons equals same element. So it's the protons that are important. We'll see how we can have different numbers of neutrons or electrons, but the protons are what determines what element we're talking about. We can look here at the periodic table, for example. The elements here in green, those are ones that are found in living systems. Oxygen there is a very tall bar. Its atomic number is 8. That means it has 8 protons. We can also look at the breakdown of what are the proportions of these elements in U. A lot of U is made out of oxygen and carbon and hydrogen and nitrogen. That's 95+% of what living things are made out of. Then we have some of these other elements here, calcium, phosphorus, etc. And then we get into these really sort of low level ones, the trace elements. They might be important, but at maybe very low levels. All right, so let's circle back to this idea of atomic structure. Here is, again, a cartoon of our atom, what it might look like. This one is helium, specifically. It has 2 protons, so that tells us that it's helium. It's got 2 electrons swirling around the nucleus, and in this case, this also has 2 neutrons there in the nucleus. So what are some other things we need to pay attention to? An element's mass number is also important. So here in our example below is sodium. And in this case, we have sodium, and its mass number in this particular one is 23. And that 23 is the number of protons plus the number of neutrons. So in this case, 23. But sodium's atomic number, that's the number of protons, that's 11. So that's what determines its sodium. So you might say, well, how many neutrons does this have? You can just subtract that mass number or subtract the atomic number from the mass number, and you get 12. So sodium has 12 neutrons. So you can change up that number of neutrons and still have the same element, remember, because protons are the thing that matters, that determines what element it is. And when you change up those numbers of neutrons, that is giving you a different isotope. So this is, again, I'd expect a term you've probably heard at some point, isotopes. Isotopes just differ in their numbers of neutrons. Radioactive isotopes are particularly interesting and maybe useful in research of all kinds because they decay spontaneously. So some isotopes, like here we have the carbon examples in this figure below, carbon-12 stable, carbon-13 stable, but uncommon, and then carbon-14 radioactive, very unstable. So this will decay spontaneously. Now, radioactive elements can be very useful in research. And so there's many potential applications. You probably heard of radiocarbon dating, for example. So dating fossils or artifacts. You can put a window of time on how old it is by the proportion of remaining radioactive carbon. So in biochemistry or in medical research, you can trace atoms through metabolic processes. So feed cell culture or potentially put into a patient a certain radioactive compound and see where it ends up by the presence of that radioactivity wherever in the cells or in the tissues. Diagnosing medical disorders. So here in this image in the bottom right, we see a cancerous throat tissue, basically cancerous tumor there. This patient was given radio-labeled glucose. So cancer cells are highly metabolically active, so they'll take up a lot of that glucose. In this case, it's radio-labeled, and so that's where you're going to see a higher concentration of that radiation, so you can track where it goes. Of course, there's a downside to radiation too, so the decay of isotopes can cause damage to DNA, for example. That could potentially lead to cancer. So at high enough levels or prolonged hits with radiation, that can have negative consequences. Let's talk more about atomic structure, but now we're thinking a little more about the electrons. So neutral atoms would have the same number of protons and electrons. And if you think about it, this makes sense. If you have sort of a +6 charge of protons to even that out, so to speak, you would need 6 electrons. So 6 negative charges, 6 positive charges equals net 0. So those would be neutral atoms. Ions, on the other hand, have charges to them. And we can see either cations. Those are atoms that have, or really compounds, that have more protons and electrons. So that you end up with a net positive charge. So you don't have enough electrons to cancel out those positives. Anions is the opposite. You end up with more electrons than you have protons, and therefore you have a net negative charge. So where are the electrons when we're looking at an atom? They are around the nucleus, like we've seen before, the swirling electrons in those cartoons, and they occupy what are called orbitals. And so this is where they spend most of their time, in this region around the atom, in this region called an orbital, and there's different orbitals where you'd find electrons. But this is all happening in a very dynamic way, but this is where they're spending most of the time. And a really important point is you've got these orbitals like orbits around the nucleus, but they could be in different orbitals. So as we'll see in a second, the further out you are from the nucleus, the more potential energy that electron has. Think of it as having more distance to fall back down to a lower orbital. It has potential energy. And this is really important for understanding how energy moves around in biochemical systems and living things, is this idea that an atom or a compound can absorb energy. When it absorbs energy, electrons can then move up to higher orbitals, and then they have a higher potential energy. And then when they move down to lower orbitals, that energy can be released. In this picture, it's saying energy lost, but that energy can be harvested to do work, as we'll see. So this is really important. So that where these orbitals are, and where those electrons are in those orbitals is really key. So the further out you are, the more potential energy you have. For now, one of the things we want to pay attention to is what those electrons can mean in terms of consequences of chemical bonding. And so the electrons in the outer orbital of an atom are really important for that. And these are called the valence electrons. These are what determine the really chemical properties of that atom and whether that atom can form a chemical bond with something else, or how many chemical bonds they could form. So if we look at an example here of a couple of different atoms, we have argon in the cartoon here. In the central orbital, or the innermost orbital, better term, you can occupy 2 electrons. In the next orbital, you have 8. In the outer orbital, in this case, the next third orbital out here, you have another 8. And so this idea where you get beyond the first inner orbital, where you can have 2 electrons, you tend to have 8s. The second and third orbital can occupy 8 electrons. So a good rule of thumb in living things is, well, 1, atoms tend to establish completely full outer energy levels. We're talking about electrons here, and usually that means 8. And so that's why we call it this octet rule. Lithium has the filled inner orbital. There's 2 electrons, and it has this 1 additional electron. So lithium is going to be very reactive because it is, you can think of it sort of missing, quote unquote, 7 electrons in this next orbital out. Sodium, same deal. It has the second orbital filled, but it's that third orbital where it only has the one electron. So very reactive. So if you have not a very full outer orbital, you're going to be more reactive generally than something like argon that has all those electrons filling that outer orbital. So this can help us determine what chemical bonding might take place with these different atoms. Let's go harken back to our periodic table. This is really narrowing down that table to some of the elements that are important in living things, and a few others smattered in here just to fill out this part of the table. But if you look down the columns here, you'll notice, for example, hydrogen, lithium, sodium. Here, these all have a single electron in their outer orbital. We go to the next one over, there's 2 electrons in their outer orbital, and you can work your way across. You can also look along the rows of this table and see this first row are elements that only have that inner shell of electrons used. The second row are atoms that have the second orbital used by various numbers of electrons, and so on. So where these things lie in the periodic table can tell you about how many electrons are in that outer shell and how many electron shells that we're looking at. And again, this is a simplified, boiled down version of that periodic table. Things get a little more complicated as we get further down the table, but for the things that are important for living things, These are the key features to pay attention to. So how about that idea of reactivity? Look at helium here, like we saw with argon before. It's got 2 protons and then 2 electrons, so it's neutral, and that outer orbital is filled with 2 for that inner orbital here. And it's also the outer orbital in this case. Whereas nitrogen, it has 7 electrons, so 2 to fill the inner orbital, and then 5 to put in that next orbital out. So you're sort of quote unquote missing 3 electrons to make up that 8. So nitrogen is fairly reactive. It could form, as we'll see, by sharing these electrons, 3 covalent chemical bonds. And so this is going to determine the properties of these different elements. So let's start putting these pieces together now. And we can introduce a couple of terms, which probably sound familiar, but here are the sort of formal definitions. Molecules are groups of atoms held together in a stable association. So you've got more than one atom. And so this could be, say, oxygen, for example. That's O2. You've got 2 oxygen atoms. That makes a molecule. So you've got 2 things there. How about compounds? They're molecules containing more than one type of element. So compounds are a subset of molecules, right? So the example I just gave you of oxygen, that's O2. That's not a compound because it's only got oxygen. It's got 2 of them, but you need something else to make it a compound. You need a mix of different elements. And then, as I've alluded to before, these things are held together by chemical bonds. So let's look at those. And we'll start with covalent bonds. And I'll go through these really also in order of strength as well. So here we have the sharing of valence electrons. So covalent bond strength can depend also on how many electrons are shared. So a single bond where you have 1 pair of electrons shared. A double bond is stronger. That's 2 pairs of electrons shared. You can imagine because there's more sharing, it's going to be a tighter association. Triple bond, 3 pairs of electrons shared. And that's illustrated here. H2 hydrogen, there's a pair of electrons shared. O2, that example I've already given you, there's 2 electrons shared, so there's a double bond, so 2 pairs of electrons. And nitrogen, that's a triple bond. And if you now look back at each one of these, each hydrogen in the first image there, they now have a full electron shell, 2 each, because they're sharing them. Same with oxygen now. They need 8 in this outer. An individual atom would only have 6. But because now they're sharing 2 each, this oxygen atom has 8, 1, 2, 3, 4, 5, 6, 7, 8, and ditto over here. And then the same logic applies with nitrogen. There are all kinds of ways to show these associations. So let's take water, which we'll see many different times. Here is an electron distribution diagram like we've been looking at. So you can count up the electrons here. Now oxygen has got 8. It's sharing a couple with 1 from each of these hydrogens. And the hydrogens now have 2 in their outer orbitals. We can see that here. We can use these Lewis dot structure formulas here showing the 8 electrons around oxygen and the 2 each shared with hydrogen. Or the structural formula here, this is more common. Oxygen with the 2 hydrogens attached. This implies you already know about the reasons behind it, the sharing of these electrons. And then these space-filling models are common as well. So what's happening in these covalent bonds? Let's look more closely at hydrogen. So here they are, 2 hydrogen atoms. They're 1 electron whizzing around in that 1 orbital. As they get closer, that electron from hydrogen A here is being attracted to the nucleus of that other hydrogen. So you see this clouds kind of extending a little bit toward each other. The negative electrons are being drawn a little bit to that other nucleus. Then you form this stable association. Now this is more stable because the outer electron shell is filled, and those electrons are really being shared between the 2 nuclei. So how does that sharing work? It's not always equal. So with hydrogen, two hydrogens, they have the same nucleus because they're hydrogens, and you have equal sharing of electrons, but that doesn't always work out like that. And so differences in electronegativity, that's how the affinity that different atoms have for electrons. So if you have one that is kind of an electron hog, even if they're sharing electrons, one is going to kind of hog the electrons more towards its nucleus than the other one. And so these differences determine these two different kinds of covalent bonds. Both are sharing of electrons, but in non-polar bonds, there's equal sharing, and in polar covalent bonds, there's unequal sharing. So if we look at the examples on this slide, in the top we have the non-polar covalent bonds, and like hydrogen, like we saw before, each nucleus has the same electronegativity, so one isn't hogging the electrons from the other, and you have a pretty even distribution of that electron sharing. In this example in B here, a polar covalent bond, HCl, you have chlorine, which is very much an electron hog, very electronegative, and so the electrons are drawn toward that chlorine nucleus and away from hydrogen, and this creates a polar bond because, like we see in this example of OH down here in the bottom panel, you have exposed that hydrogen nucleus partly over here. So it leaves this end of this bonded pair with a slightly positive charge. And this one, in this case, oxygen, that's hogging the electrons, it has a slightly negative charge. So this creates a polarity. And so that's why this is called a polar bond. And this is not happening with our non-polar bonds where there's equal sharing. How do we know who is the electron hog and who is less so? We can look at this electronegativity table and determine whether a bond is going to be polar or non-polar depending on the difference. The rule of thumb is that the bond is going to be polar if the difference in electronegativity is 0.5 or greater. And there's ones that are probably borderline, and then it comes down to empirical evidence whether that's a polar bond or not. But let's take that example of oxygen and hydrogen. Look at the difference between those two, 3.5 and 2.1, definitely more than 0.5. And there we see oxygen hogging the electrons away from hydrogen, and we've created a polar covalent bond. Okay, how about ionic bonds, which are, in living systems at least, almost always less strong than covalent bonds. What's happening with ionic bonds here? Remember, there's some atoms, elements, that have just, say, one electron in that outer shell, so they're reactive and not stable in that way. So sodium is an example. Chlorine is the opposite. It has almost 8 electrons in its outer shell. So when sodium and chlorine get together, what happens is sodium loses an electron to chlorine. Now sodium has a full outer shell, because that what was its outer shell is now empty, and the second shell has 8 electrons. Chlorine has picked up another electron, and therefore now it's more stable, but they're both ions. So sodium is a positive, it's a cation. Chlorine is an anion. And so now what's holding them together? Just negative and positive charges. So they stick together. Think of like magnets. They're not sharing electrons, so this isn't as good as a covalent bond, but they stick together. This is salt, right? So how do we know that maybe this isn't as strong as a covalent bond? What happens when you put salt in water? It dissolves. These things will start to break away. Water will break this up. We'll see that later. But that's how ionic bonds work. It's just these ions sticking together. Here we start to get in what we would call weak interactions. That does not mean they're not incredibly important. So together these could be as important as any other chemical bond. And a good example here are hydrogen bonds. So a hydrogen bond is formed when you have that, say, a slightly positive exposed hydrogen in one compound that is attracted to the negative charge of nitrogen or oxygen in another compound. So we see in this little cartoon here this hydrogen of water with its slightly positive charge, because this is a polar covalent bond, is attracted. It's not bonded, but it's attracted. It's slightly negative, slightly slightly negative, slightly positive, kind of like ionic bonding. So there's drawn close, but it's not as strong as an ionic bond because these are just weak. These delta plus, delta negative, these are weak charges, but it draws them together. So these can be very, very important. Other kinds of weak interactions called van der Waals forces or van der Waals interactions. And these are kind of even more sort of nebulous than, say, the hydrogen bond, where you have sort of areas on a surface of slightly positive, slightly negative charges. Those kind of line up. That actually can provide a lot of strength. An example would be of this lizard climbing up a wall. They've got a lot of surface area on their little fingers there. And just the slightly positive, negative charges on those surfaces interacting with, say, the wall, they're able to climb up. And so they're not suctioned onto there, but it's these very weak interactions that come together and make a very strong force to allow these lizards to climb up. All right, that ends this video lecture. In the next one, we'll talk about basic chemical reactions, water, and pH.